Professor Qing V. Wang, I invite you to first to share your research experience with us. So Wang-san, floor is yours.
Okay, thanks. Thank you, Amit and Hidaki Sensei to introduce. Yeah, very happy here. Hi everyone, I'm Qing Yuan from Institute of Applied Ecology, Chinese Academy of Sciences, China. I'm interested in canopy solar spectral ecology. 2 years ago, I worked in forestry and forest products research in Japan, and I carried out a GSPS project collaborating with doctors Hiloko Kulakawa and Marshall Robertson. Today, we're happy to have this opportunity to report our research advance for the photodegradation. My report title is Anaerobic photodegradation. Solar radiation is accelerating carbon cycle process of temperate forest ecosystems. As we know, climate change is the greatest challenge for humanity in the current century. The evidence is becoming stronger and stronger. For example, NOAA has identified January 2020 is the hottest January on record for the Earth. One important reason is increasing CO2 level, which hit a climate milestone last year. The first time over 415 ppm. So it pushed us to understand well the global carbon cycle and the mechanism of the key process. Among all processes, data decomposition is a critical step for carbon cycling among ecosystems and between the biosphere, atmosphere. Data decomposition generally is controlled by climate, for example, temperature and rainfall, light quality, such as carbon, nitrogen, lignin, and decomposers. However, traditional models based on this triangle always underestimate carbon turnover rate, especially in dry land. In such a hot, dry environment, model decay rate is much lower than the budget value. The big reason is a stronger irradiance is not included in the model. Solar radiation with different spectral composition does not only provide energy for photosynthetic carbon input, but also accelerated carbon backflow to the atmosphere. For photodegradation mechanisms, in brief, sunlight can directly break down the complex macromolecules, such as lignin, into small organic compounds. And this process releases the greenhouse gas. Small organic compounds can indirectly benefit microbial decomposition. The former process is called as photochemical mineralization, and the later as photofossilization or photopriming. Photodegradation has been discovered in marine ecosystem in 1989 and generally identified in dry land since 26. Austin group has identified sunlight increase the decay rate by 30% compared with darker condition. Recently, they find that photodegradation had a spectral dependence. For example, short wavelength visible light has a greater contribution than UV radiation in a semi-arid ecosystem. As we know, sunlight can be found anywhere on the land. However, the significance of photodegradation in massive ecosystems is much less understood, particularly in dynamic light environment, such as temperate forest. Temperate forests mainly depend determines the strength of the terrestrial encompassing. Solar radiation reaching the forest floor varies greatly both in the quantity and the quality here because of canopy phenology and gap disturbance. So this layer is always exposed to changing light environment. However, until now, nature is knowing about the importance of photodegradation in temperate ecosystems. It limits us our ability to predict how climate and land use changes will affect the ecosystem processes. Our hypothesis is that photodegradation accelerates the litter decomposition in temperate forest ecosystem, but it would be greater in the gap than in the understory, where it's really dark in summer. In the gap, photodegradation would be driven by short wavelengths with light, considering the formal mechanisms. So the variation in photodegradation among species could be predicted by initial litter traits, for example, lignin and phenolic compounds due to their optical properties. We carry out a larger spectral attenuation experiment in Okawa Forest Reserve in central Japan, one of Ilter EAP members. This forest is a cold temperate deciduous forest dominated by oak and beech species. We set up 2 plots, one in understory and another one in the gap. The gap is clean cut within 1 year before the experiment. So 2 plots have similar environmental conditions. We made 6 spectral attenuation treatments using different phenotypes to make the little boxes. The treatments include transmitting 4 spectrum as control, and remove UV, remove UV blue, remove UVB, remove UV, remove UV blue, and remove UV blue green, and the last one is dark. We select 12 widespread litter species and determine 12 common initial litter traits. This is the litter box prepare the process image. Finally, we made 2,400 litter box in total. Litter boxes was placed in April before canopy flush and collect 4 times from June to December, according to the canopy phenology. Totally, 2 plots covered around 500 square meters. Okay, let's see the result. First, the temperature. You can see temperature was similar among treatments in each plot. Soil moisture also has no significant difference among filter treatment. But for cumulative solar irradiance, for each spectral range was much lower in understory than in the gap due to the canopy closure in the summer. Accordingly, a later image looks similar among treatment in the understory, but in the gap, much loss appear in the former 3 treatments. These 3 treatments, all these 3 treatments included blue light. Once we attenuate the blue light from later 3 treatments, we can see there's not much change in litter. Such a variation is well reflected by the mass loss figures. So it suggests that solar irradiance accelerated the mass loss in the gap. Further, we examine photodegradation effect on mass loss by calculating the response ratio to the spectral contrast. For example, if the value increase above 1, it means Leadter decomposition is faster. We found the photodegradation in the understory was positive, responding to UVB over the first 15 days. But it's because UVB wavelength is much shorter and easily penetrated into the forest floor. But for the gap where sunlight is strong, the contribution of full spectrum and blue light to photodegradation was greater than others over the whole period. Similarly, understory photodegradation on decay rate was similar among different spectral range except for UVB radiation, but accelerated the decay rate by 120%. Interestingly, there 75% of such an increase is contributed by blue light. However, such a photodegradation effect was always ignored in previous study and models. We already knew positive photodegradation effects on Leadter decomposition, but how much it contributes to the carbon loss at the ecosystem level when we consider the gap creation. To answer this question, we made a model simulation and found photodegradation increased carbon flow back to the atom by up to 63%, compared with the continued forest understory. It suggests that solar radiation is accelerating carbon cycle process of temperature forest ecosystems. We identified initial lignin and phenolic compounds as the strongest predictors of photodegradation. The apple is initial lignin with photodegradation. High lignin later was most prone to photodegradation from UVA, blue and green light in the gap lane. Interestingly, high phenolic layers in understory promote UVB photodegradation, but suppress it in the gap. It suggests that the dual role of both lignin and polyphenolic compounds affecting Leadter decomposition. Here, I would like to give a summary. Our study shows that photodegradation driven by blue light is an important driver for decomposition in massive ecosystems, not just in dryland of the world. It's broadening our knowledge that photodegradation is ubiquitous, contributing driver of decomposition over biomes. It may explain why carbon cycling is underestimated by the traditional models. Such a significance also has received increased attention from a UNEP environmental effects assessment panel. Its update 2019 has described a major effect of global change on carbon cycling is after the exposure of natural organic matter to solar radiation. In update 2020, it directly points out that photodegradation can partly explain the missing carbon loss in terrestrial ecosystems, leading to an estimation of global carbon emissions. Fortunately, I have been invited to some previous studies and made a concept framework by separating massive and dry ecosystems. In order to find the generalization, we also did a meta-analysis. However, photodegradation and its dry spectral range varied greatly in temperature terrestrial ecosystems. We find the reason is about heterogeneous methodology, data materials, and environment. So there's an urgent need to carry out the large secure standards long-term monitoring network. Accordingly, we established a regional secure decomposition network in 2020, but only in China due to the pandemic events. This network, we select 50 plots, use the 12,000 standards Leadbox, and cover most environmental gradients and visiting tips. So I will be very happy to share us the results once it is finished. So for more information about the solar spectral ecology, you are welcome to visit or join UV4 Plus. Finally, I have to thank many research institutes and grants for great support to this study. It's all my talk today. Thank you very much for your attention. Due to a time limit, I did not introduce it more detailed. So please give any comments or suggestions on our study. Thanks.
Yeah, thank you, Professor Wang. Very informative presentation. And I learned a lot of things, which is new for me also. So I think this is the time to ask questions. So yeah, please, you can put your questions on Q&A chat box, and or you can raise hand. So you can ask directly to Professor Wang if you have any questions. Okay, so I think Professor Hideki want to know something. So Professor Hideki.
Yeah. During the waiting, the question from the participants. Thanks, Wan-san. It's very interesting. It's a very nice talk. And then I'm impressed that your litter box experiment in the Ogawa forest. And then it seemed that that box can cut several wavelengths of the solar radiations for your manipulation studies. So my question is that, so I'm interested in that the current mechanisms, what happened due to the change of radiations. So about the abiotic impact and microbial impact and chemical impact. So using this litter box, are there any change of the micro temperature and moisture in the box due to the change of the wavelengths? And also, did you find any actual change of organic matter compositions in the litter due to the change of radiations? Or did you find any microbial biomass change by the altered radiations by this treatment? So any thoughts or comments?
Yeah, it's good questions. So many, and also new for us. It's my first time to make such kind of Leadbox. It's because of we have to make the air flow inside. Otherwise, there will be molds or some other how to say the microbial factors. The temperature we show on the slide here, the temperature was recorded in the Leadbox, not outside. So you can see here it's the full spectrum and dark 6 kind of Leadbox. In the end story, they all almost overlap on one line, also in the forest gap. So I can confidently to say there's no much difference in the temperature among Leadbox. So another one, the moisture, because we can't measure the moisture inside because you know it's limited by a technical. If the water is too much, maybe the motion can't work. If we, so finally we discussed with Hilko Kulakwasa and decided to catch the soil from and the Leadbox around 5 centimeter below the Leadbox to identify the moisture effect, the decomposition rate. So finally, we can see this here. That this result we have did not get a significant effect of treatment. And the second point about microbial, yes, we really find some effects from microbial or the larger soil animals. For example, in the end story, we can see before 110 days, the color did not change too much, but later the color become much lighting and yellow. We guess it's because some lignin fungi what grows on the leaf surface, but outside in scape there's no much no such color because of the sunlight is strong. Also, the temperature fluctuation is too great, so such condition may not benefit the microbial community and the microbial decomposition. Another point is about the dark treatment. You can see the cover figure from the gap. The dark color index dark treatment. This one is how to say, not in our prediction. We thought that it should be the opposite, but now it's lost in the intermediate of figure. During we treated the Leadbox after collection, we find many animals in the dark treatment. I have an image at the end of the slide. Here is a dark treatment of Leadbox. You can see here's many Havre and dropping insect bodies. So we guess the soil animal or larger how to say fauna prefer the dark condition in the hotter and very how to say high irritants condition. So they want to find some place as a home, for example. Yeah. And the third one for the lignin or for the organic matters, actually we measured, and the lignin is really have a good response or good relationship with the spectral range, for example, blue light and UV light. But we put another this result on another paper, so I have not put here due to the time limit. Thanks.
Okay, very interesting. Thank you so much.
Thanks.
Yeah, thank you, Wang Sang. Here I found one question from Prakash Jha. In case of altitudinal variation, has gradient analysis been carried out to find out correlation, if any, with other parameters?
In case of
altitudinal variation has. Can you see the questions on Q&A?
Yeah, this is good question. It's not included our study here. We just carry out the same position or same altitude. But for the altitudinal variation, I think increase the temperature decrease, but UVB or solar irradiance is increasing. So I think there's a trade-off. I'm not sure which will dominate the result. Yeah.
Thank you, Wang San. I think, yeah. So anyone, if, okay. So I think Dr. James also can ask if he is interested.
Yeah, you're welcome.
Yeah. May I ask you, like, I'm wondering, is there any limitation of such study or such research?
Limitation.
In case of like the study site is very small, I guess.
You mean the Leadbox?
Yes. And
Yeah,
the study site is also very short, and I think is.
Yeah. In order to make such a Leadbox, I have such a lot of stores in Japan to find the biggest store. But finally, the diameter of this store is about 1.3 centimeter. The size is 20 and 20 centimeter. So of course, larger is better to make how to say the leisure much closer to the natural status. I think, yeah.
Yeah. Thank you.
Yeah, the process is very hard. Actually, we use four persons consuming 3 months to make such a Leadbox.
Yeah, that is my concern. How time consuming.
Yeah. If you watch it carefully, you can see each filter we have makes a hole allow the rainfall inside the Leadbox. So this is also very hard. Not we can't push the hole on for example 10 or 100 pieces, just maybe 3 or 5 pieces to push the filter. So it's very hard.
I see.
Here, yeah.
Yeah, thank you for your clarification. Yeah, I think Dr. James Weldon also want to know something. He's
please.
Yeah.
Yeah, it's just one of your slides. You mentioned that this raises the question of the urgent need for a kind of widespread standardized monitoring. And since we're in a forum hosted by ILTER, the obvious question, I guess, is what role do you see that the ILTER network could play in facilitating this, if any?
Yeah, actually now the plot, how to say, show the plot. Yeah, this plot at the beginning last year, I have no confidence to set up so many. I just maybe 10 or less than 20 at most. But finally, many friends and many researchers were interested in such a project, so they, how to say, they support us freely to set the samples and plot in their own station or near their plot. So how to say, at this moment, this plot or this network not belong to any kind of national or international network. If it's possible, I'm really happy to share or join Ilter or some other network, yeah, to find a more generalization of photodegradation in different ecosystems. And this network, we plan to run 3 years. Now, 1 year already passed. We started in the last October. Now, how to say, for this 50 plots, we have organized 38 groups, research groups, to manage and make more deep research on it, not just only decomposition rate. We also to separate different specific teams. For example, the lignin structure and microbial mechanisms and gas emissions, yeah, along different gradients or different vegetation types from forest to desert.
Yeah, thank you, Professor Wang. I think it is clear to me, to everyone also. I think the time is very limited. I think I have to move and invite our second great speaker, Dr. James Walden. So, Dr. James Walden, floor is yours. You can share your present file.
Okay.
Yeah, please.
Yes, thank you.
Thank you, Professor.
Okay, so I hope you can see my screen. All good? Great.
Yeah, great.
Yeah. So thanks for the opportunity to present this. And I'm going to be talking about a paper that I wrote together with my colleague at the Swedish University of Agricultural Sciences, Ulf Grandin, and just published earlier this year in The Lichenologist, entitled Weak Recovery of Epiphytic Lichen Communities in Sweden Over 20 Years of Rapid Air Pollution Decline. That QR code will let you download it. It's an open access paper, but I'll show that again in the end slide, no rush. So what we're looking at here is primarily epiphytic lichens. There's a nice leafy example here from a common species in Sweden, Lobaria pulmonaria. And one of the qualities of epiphytic lichens is that they're well known to be good indicators of air quality. The reason for this is that they're very susceptible to taking up mineral nutrients, including air pollutants, because of their basic physiology. They can take up nutrients across their entire surface. They also have a slow growth rate and are often exposed to air pollution because of their growth habits on exposed surfaces. And for a given concentration of sulfur, they will absorb more of it than a vascular plant in the same environment. The other part of the title is, of course, atmospheric pollutants. And this encompasses a range of compounds emitted by various sources which have various effects. The parts we're interested in here are highlighted in yellow. So we're looking at the impacts in forests, so terrestrial ecosystems of acidification and eutrophication. And the compounds we're really interested in are sulfur and nitrogen compounds in this context. And this, of course, became a very important issue due to the so-called acid rain that was prevalent in Europe, at least in the 1970s and 1980s, which prompted international agreement and international action to cut emissions. You can see here a forest that's being killed by acid rain in the Czech Republic. Also lakes, of course, suffered badly from this phenomenon. So in many ways, this has been something of a success story. The emissions of sulfur dioxide peaked in Europe in the early 1980s. And then because of this concerted action have declined steeply since then. Nitrogen oxide emissions peaked around 1990 and have declined somewhat, but not nearly as much as sulfur. And ammonia, again, some decline, but really far less drastic than what we see in sulfur. So yeah, we have this strong declining trend, particularly in sulfur, less so in nitrogen. So that was the European context. How does it look in Sweden? So we can see a time series from left to right here showing deposition in Sweden. So we can see that it's declined over time. The blue areas have expanded from left to right as time progresses. The red areas become smaller or disappear. Another phenomenon we can see is this north-south gradient. So southern Sweden has had always higher levels of deposition than the north, and particularly this area of the west coast, which has always had the highest levels of deposition. So even though they have declined, there is still this gradient. So the data we're using here come from the ICP Integrated Monitoring Network, And there are 4 sites in Sweden which are distributed along this distribution gradient of atmospheric pollutants. So we have one site, Gårdsjön, on the west coast, which is this high pollution area. Another one in the far north, Gamtratten, which is considered to be a pristine site, which has always had very low levels of deposition. So we've been running this monitoring scheme for many years now, and this study incorporates 20 years of data for each of the sites, where lichen monitoring has been conducted using a standardized methodology throughout the period. The selection of trees is randomized, but spruce is dominant in the areas that the site's located in, so it's also dominant in the monitoring program. One of the sites had extensive damage from storm and bark beetle outbreak from 2005 onwards on a border. So some of the results from Arnaboda have become a little strange because of these disturbances. So it becomes hard to then disentangle the effect of the disturbances as opposed to the effects of the deposition levels, but we'll come to that. So we can see here the same trend that we saw on the national map of Sweden reflected in the data from our monitoring sites. One of the advantages of these sites, integrated monitoring, means that there are data for biodiversity and deposition and a range of other variables, all taken consistently from the same sites. So we can see the same gradient in deposition with Gamtratten, our site in the far north, being at the lowest level, higher levels, highest at the west coast site in the south, Gårdsjön. But again, we can see this declining trend in atmospheric pollutant deposition almost everywhere and in almost every case. So what were we hoping to find or expecting to find? So we know that because of years of extended deposition of atmospheric pollutants, that the lichen community at these sites would have been depleted at the start of the monitoring period. So we would expect, with the continued decline, that these should show a recovery. While the lichen in this northern pristine site, we would expect to not show temporal trends, as it should not have been strongly affected. We noted that the levels of sulfur have declined more sharply than nitrogen deposition, so we'd expect the sulfur-sensitive species in the lichen community to increase at the polluted sites, while maybe not showing so much response for the nitrogen deposition, the nitrogen-sensitive species. How did we look at this? Okay, so for each tree, we made a community weighted mean value for the species present and their tolerances for air pollutions based on these sensitivity values that were published a while ago by Hultengren and colleagues. So a higher value in this Hultengren sensitivity index means it's less tolerant to sulfur. So these are less acid tolerant species. And again, the same for nitrogen using values given in Wirth et al. So these again range from oligotrophic species to eutrophic, and we did a community weighted mean. We also calculated Shannon diversity indices. We then broke down the beta diversity into the components of turnover and nestedness. The idea here being that if the change in beta diversity is driven or dominated by nestedness processes, then the species poor sites are a strict subset of the species rich, and this implies an increased homogenization of the vegetation community, or the lichen community in this case. Vegetation in the broadest sense, they're of course not plants. So then we looked at changes over time in these measures of diversity and the community indices for sulfur and nitrogen preference using mixed models. I won't dwell on the details here because we have so much time. To move on to the results. So sensitivity to sulfur, we can see that as we were expecting, the mean sensitivity of the community present at the most polluted site in Gårdsjön has increased over time, in line with our expectations with reduced deposition. But we also see that it's declined at Kindle and Gamtratten, which are sites with low deposition. So this was a little concerning that our supposedly pristine northern site shows decreased levels of sulfur sensitive species. Again, the Ona border site after 2005 becomes very difficult to interpret these results because of these extensive disturbances. We ended up with a very heterogeneous habitat with some pockets of intact forest and very extensive disturbed areas. So it's very hard to make conclusions from the Ona border site. In terms of nitrogen preference, we can again see that the mean value for nitrogen preference decreased at the most polluted site, Gordhuan. This is a positive signal. But we also see that it increased at Schindler, despite, again, these rather low levels of deposition, with no changes at Gamtraten and Ona border. Moving to taxonomic diversity then. So the lichen species diversity decreased at Gordhuan. Despite these decreases in deposition, we see a decline in species diversity. And again, no changes at Schindler and Gamtraten. Again, these changes seen at Ona border are hard to interpret because of the disturbances. So to summarize some of these results, we could say the most polluted site at Gordhuan, we have this increase in mean sulfur sensitivity and a decrease in nitrogen preference, which is good. But we also, looking more closely, found that there was no increase in the richness of the most sulfur sensitive species. So this mean change in the community weighted mean preference is driven by a loss of species that are tolerant to highly acidified conditions, but not an influx of the species that are highly sensitive to this to replace them. On a border, we saw no significant changes. Schindler, decrease in mean sulfur sensitivity and increase in mean nitrogen preference, which is, again, from a conservation point of view, not really what you want to see. And our pristine northern sites, again, showed a decline in sulfur sensitivity despite these very low deposition levels throughout the monitoring period. Taxonomic diversity, again, we have this decline in richness and diversity and signs of homogenization, so an increase in the proportion of beta diversity that's explained by nestedness. At Ona border, we have a decline in richness and diversity again, but yeah, storm and beetle effects make this hard to interpret. Schindler, no significant changes, and again, Gamtraten, no significant changes, which is what we expected. So we found that across our sites, levels of nitrogen and sulfur deposition have generally declined sharply. But despite this, we've only seen this quite partial recovery at Gordhuan, the most polluted site. So we have a loss of these species that are tolerant of high levels of nitrogen and sulfur deposition, but a lack of new sensitive species coming in to replace them. And even in our more pristine areas, these areas with low deposition or intermediate deposition, we've seen declines in sulfur sensitivity and a eutrophication signal, even at low levels. How can we explain this? Well, sensitive lichens can be lost quickly in periods of high deposition. That's what makes them good bioindicators. So they're very useful in the deposition increase phase. And we know that even low level deposition can have a cumulative effect on them. Another factor is that air pollution is a disturbance which, of course, isn't localized. It takes place over very large geographic areas and over long periods of time. So we would expect that the regional species pool is affected, not just at our monitoring sites, but regionally. Another factor in this context is that these monitoring sites are all nature reserves, effectively. So there's no logging, there's no forestry permitted in them. The surrounding areas are almost always production forest. There's a few nature reserves dotted around, but generally the overwhelming majority of the forest surrounding each of these sites is used for forestry, for production. So it's quite young. We know that a lot of lichen species require old growth forest, and the conditions it creates are necessary for their reproduction and survival. So these factors together, the depletion of the regional species pool and the unsuitable habitat created by forestry, would imply that there could be quite long distances for any recolonization to occur over, even now that the deposition levels are low enough to permit these sensitive species to come back. They have to come from somewhere. We know from other studies in this area that recolonization can be a very slow process. This paper found that one sensitive species has managed to recolonize only 35 meters in 9 years. And this potentially has implications for using epiphytic lichens as bioindicators during the phase of deposition decrease. The fact that they're absent doesn't, in this case, imply that the air quality is poor. The air quality has improved to an extent that they should be able to recolonize. It's other factors that seem to be coming into play to prevent this recolonization being successful. So it's just something that might need to be borne in mind when using lichens as bioindicators, as surrogates for measurement of actual levels of atmospheric pollutant deposition. And that was the end of my presentation. Thank you for your attention. Thank you for the opportunity to present. Welcome any questions or comments.
Thank you, Dr. James Walden. It is very informative presentation. I like the theme because I'm interested in air pollution and the effect on forest ecosystem. So I think I learned a lot of things, lot of ideas from your slides. So let's, if anyone have questions, so please ask using Q&A box. Yeah, I think I can ask again, Professor Hideki Shibata. So you can ask directly.
Yes, thanks, James. It's very interesting and very important topics, and I like that. And then my question is that are there any possibility to think about any other combined effect, not just sulfur and nitrogen pollutants, such like ozone concentrations or other micro elements in the atmosphere, or any combined effect, climate change, like change of temperature and precipitations, make a slower recovery of lichens even in the decreased condition of air pollutants?
Absolutely. I mean, there's one of the as I mentioned, these sites which are also part of the European Ilter network. I mean, there's a wide range of variables measured there, and there's a lot of things that could potentially have an impact here. I mean, our focus in this was the kind of what you might call the classic factors that are widely known to affect epiphytic lichens, but there could be a range of other variables that would be worth looking at, and particularly temperature changes or precipitation changes. And there's a wealth of data that would allow us to look at very many variables, including the ones you suggest. So there's for sure potential to expand the range of potential impacts.
Okay, so you show that the slow recovery, even the decrease of pollutant over 20 years. Did you see similar tendency in your region, Scandinavian region, like Norway or Finland, in the other ISP forest, if there are some lichen studies there? I'm not sure.
Yeah, there's not it's not a epiphytic lichens on standard variable measured by ICP forests, unfortunately. So there's not so many sites that measure them. They're also quite hard to identify reliably. So there isn't a wealth of data, unfortunately. It would be nice to have more sites that did include epiphytic lichens as part of their standard monitoring protocols.
Okay, thanks so much. Yeah, I know that some ILT researchers in Portugal starting with lichens as a bioindicator for air pollutants. So the IoT can potential to collaborate to compare that kind of long-term change of lichens with air pollutants change. Thank you so much, James.
It would be really interesting to look at if the same failure to recover can be seen in other areas which have now got improved air quality.
Yeah, great, thanks.
Yeah, thank you, well done. I got one question from Eleanor, maybe from Austria. Based on your result, will you still recommend the use of lichen as a bioindicator of air pollution?
I think before making general sweeping statements, I would like more results from a wider selection of habitat types. But I would say, in terms of worsening air conditions, they're of course well known to be useful in that context. The only issue we're kind of raising a little bit is this question of being a little bit cautious about them in terms of the declining phase of air pollution, because they maybe cease to correlate the presence of the lichen with the level of air pollution. Maybe that correlation breaks down when they can't recolonize. So it's just something to be aware of. I couldn't say if it's a very generalizable thing or if it's specific to our conditions, but it's something to bear in mind in your context and think about if you're going to use lichens as bioindicators, I would say.
Xiwen, please raise your questions.
Thanks, James, give us a very exciting presentation. And I have a small question about the sensitivity. How do you, or how examine the sensitivity of lichen? For example, you calculate by growth or by some other parameters. So I'm not quite sure. Yeah.
Yeah, I mean, this is based on work done by Sven Hultengren and colleagues back in the 90s. So they did extensive research on the sensitivities of different lichen species. I'd have to go back and check their methodology, exactly how they came up with that.
Okay.
But yeah, it's the Hultengren group that did that research back in the 90s.
Okay, thanks.